saw a news story yesterday where the city of Plano, I think it is, has introduced mosquito fish in their ponds and lakes there to control mosquito population.
Man, you got me on another soapbox. The issue with Zika, and I'm sure most may know or not know, Aedes aegypti and Aedes albopictus are container breeders. They don't breed in standing water hardly ever. Most Aedes species are floodwater breeders. Specifically, aegypti and albopictus, called stegomyia collectively. They breed in containers. So garbage in the backyard, a kid's dump truck that's left in the sandbox, if it can collect an ounce of water, you'll have mosquitoes breeding in it quickly. Mosquito fish, I mean, unless you put one in every flower pot or vase, they're going to do no good. They're going to be great for St. Louis encephalitis vectors, West Nile virus vectors, Western equine encephalitis virus vectors. But I don't believe they're going to do any good for Zika virus. And that's the big concern right now.
You make any questions? Yes, sir.
I'd like to start out with a definitional question. How long does something be there before it stops being invasive and becomes indigenous? And when does it get its green card?
We'll build a wall.
I mean, for instance, is the grackle still considered invasive? I mean, I know it's a slippery slope question, but I'm curious.
I don't know that a species ever truly crosses that line between being invasive and actually becoming indigenous to that area. I think it maybe becomes what we would consider a common species for that particular area, but I don't know that it really, at least in my mind, should ever be defined as an indigenous species.
Well, doesn't that then lead to this kind of sort of romantic sense of the Garden of Eden, the original purity of something that's been defiled, and it doesn't get into more policy than actual, because everything's we're invasive.
Invasive species. So I went to a little liberal arts college for my undergrad at Southwestern University and majored in biology, but I minored in sociology. And that really was what led me to study invasive species, because I think there is much of a social human problem and construct as they are a biological thing that we can put our fingers on. And so that's why I think in that diagram that I showed you, the last step there is those ecological and economic harm. And that's going to be defined by people. And so I think I think what you're getting at is that there is a sociological component to it, a human value component to it, and a policy component to it too.
I mean, at some point, the cultural carrying capacity of a species is going to be much higher than maybe it was initially when that species first became a nuisance and recognized as an invasive. As society gets used to having that species around, the interactions with that species and the ecology of the area and the rest of the ecosystem become more regular and more natural. The cultural carrying capacity, the amount of individuals that society will allow and that society can deal with, that carrying capacity will get higher.
Can I comment? Sure. I'm not an ecologist, but I think about feral pigs, feral swine. They've been here a long time, since the mid to late 19th century. Johnson grass that you see growing on all the ditches and fields around here, that was introduced, and it becomes invasive. Feral hogs were introduced and became invasive for game hunts. I don't know that. I've never thought about that. When can we quit calling them invasive? Feral hogs, we don't want to, because they are out of control. They found a niche.
Wild horses and crows are pretty bad for the landscape. They're federally protected. They love them.
I'd consider the pigeons at the administration building invasive at this point. You had a question, sir?
In general, do you think the invasiveness of species increases with climate variability or will decrease? because obviously species invade and home range changes and they migrate. But as we change the variability of our climate more and more, how do we anticipate the invasive characteristics of species? Because obviously, you talk about RNK selection and all those reproductive components of species, they have to come into play also as part of this.
We know for sure that the ranges of invasive species or the potential invasible range is going to change for sure. And so I think that's why this is a really appropriate, this was a really appropriate topic for CSC, right? Biological invasions, global climate change are both really important interacting elements of global change.
That's a tough.
I think adapt and establish is
Yeah.
what's going to happen.
That's a good question because if you look at, for example, wildfire, the common idea is that wildfires can increase. But that's only in the Rockies because that's where all the studies are. Other places might decrease. So it'd be interesting to see a global study of that. Obviously, generalists will be better.
Right, generalists would do better.
Yeah.
Not as well, interesting. One of the reasons I bring this up is there was a paper that was just published about 3 or 4 years ago about one of the largest increasing aspects of invasive species worldwide right now that we don't pay attention to are pathogenic fungi, and that they far outnumber bacteria and viruses almost 10-to-1 in terms of what's happening globally. Part of it's airplane traffic. Part of it's just the fact that as climate changes, these organisms are able to adapt more and more to what, and so they're entering new habitats, and so it's and a lot of them are all plant pathogens. We don't pay much attention to invasive aspects of organisms on plants, but we're changing all the time. I mean, sorghum bunt is one of the ones now that everybody's doing.
You mentioned adaptability in your response there. What's really interesting to me about invasions is that back in the 50s and 60s, when Charles Elton, the father of invasion biology, was writing about invasive species, they were interesting because invasions were rare events. Remember, each one of those steps is a funnel. Yet they do so well. We expect that if they're rare events, there should be a really genetically not a diverse population, really crummy population. How are they doing so well? And as we explored more and more, we're finding that actually invasive populations can sometimes be more diverse than the native range populations, because we get things like admixture where different genotypes from across the range are coming in contact with one another that wouldn't have ever come in contact in the native range. We are seeing those opportunities for adaptation in new niches and these novel environmental systems. And so this adaptability and evolution in invasive populations is a really interesting and important area of research.
Well, I thought that one of the things that we have, obviously in the last 300 years as human populations have increased and climate variability has increased, what we've done is we've selected for those series of traits in all the natural populations, whether we realize or not, as a consequence of us acting as part of that filter.
Any other questions? Yes, sir.
Do we know the climatic limits for the nasty mosquitoes? I mean, is it the freeze line? Is it?
We do, and we're really above it. But now you're going to be talking about artificial islands that we create in urban areas. You know, on the high plains here in Lubbock, a great example, and I've talked to some folks this morning about, we have trees, we have non-native trees, grasses, shrubs, vegetation, irrigate lawns, flower pots in our backyard. So we've created our ability for for Egypti and Alopecia to establish themselves and exist. And I don't know if you've seen the CDC distribution map for Alopecia and Egypti, the most recent one. They did a big survey in January and February of everybody in the country that works with mosquitoes. Said, hey, if you've ever caught them, when did you catch them? Numbers, all of that. Everybody responded back. They published a map last week. You know, the whole southern half of the United States, but it goes into Wisconsin, most of New England. They've been found, and so it's a very adaptive species, particularly Alopecia. For something that was introduced into North America in the 70s, early 70s, in used cars. It's done very well, kind of like Zoomer, they go hand in hand.
Anybody else got any questions? Yes, sir.
So what do you think with the genetically engineered mosquitoes they had trial with in Brazil? And
The
you want to
Oxitec mosquitoes, I think if you're, I'm sure you're all histories or historians on disease control, but you know, the primary screwworm was a major threat on the southern Mexican border, southern Texas border, post-southern U.S. border, at the turn of the 20th century. They sterilized males, released millions and millions of them out of Kerrville area, where the head of operations were. They basically eliminated the problem with primary screwworms in livestock. I think the potential for the Oxitec genetically modified mosquitoes is huge if they can produce enough. And one of the problems is primary screwworms were confined and were a regional issue. You know, maybe a couple of million square miles where they occur. When you talk about Egyptine Alpictus, we're talking about near cosmopolitan distributions. And so getting enough mosquitoes and releasing enough, I think, is going to be tough. But it's very exciting. I mean, for local areas.
You got a question, sir?
I have a question for Dr. Barnes. Like, how do you use the data you generate from the environmental DNA in relation to invasive species? Like, there's a big, huge gene pool over there, and like you get a small fraction of the pool from the environmental DNA, and it can be related to some native species itself. So, how do you analyze the data you generate from there?
Right now, the field of people looking for DNA of macrobial species is fairly young, and so we're developing that every day. Right now, we tend to target mitochondrial markers, chloroplast markers, things that are going to be very species diagnostic and differ between species. We don't do, I haven't seen anyone successfully do any population genetics with eDNA because it's this big gross mixture of all sorts of DNA that's in low quantities and low quality. I think that's an exciting frontier if we can figure out how to do it. I think right now some of the big questions that we're trying to figure out are how long does the DNA stick around? Does the amount of DNA in the environment relate to the amount of biomass? What kind of environmental factors in this ecology of eDNA framework influence that amount of DNA, and what kind of DNA is out there? So exciting possibility, but we're definitely not there yet.
So it's more like a metagenomic analysis.
So that's an exciting frontier too. I've got a student that's going to be doing some metagenomics this summer. We're moving from, is species X here, our zebra mussels here, our Asian carp here, to saying which species are here, where we take a total environmental DNA sample, amplify using generic plant primers or something like that, a fish family or a group of species, and then sequence them and say which organisms did we find, and that's a really exciting frontier too.
And what about the bacteria present in your sample, because those are so ubiquitous?
This is one of the reasons why we have to bring for that workshop, we're trying to get microbial ecologists and hydrologists and people from diverse backgrounds all in the same room, because of course the microbial people have been doing this, right? You can't culture most bacteria in the lab. So what do? They go out and they look at the essentially environmental DNA. The first person to use the term environmental DNA was a guy named Ogram in 1984, and he was sampling sea vents and the bacterial communities there. So we are building on the shoulders of giants. We need a lot of interdisciplinary collaboration to make sure we're not reinventing the wheel, and to make sure we're combining useful information from all these different fields.
All right, I think we need to go ahead and wrap up today. I want to thank our three panelists. Let's give them another round of applause. be it for this semester. We'll look forward to seeing you all back in the fall. Thanks a lot.
So I was